Hey everyone, welcome to my YouTube channel, and today, as a part of microbiology series, we will be talking about bacterial staining. Um, Visualizing microorganisms would be very difficult if they are in the living state, and because they are so minute, okay, and transparent and practically colorless, when suspended in like a medium, like aqueous medium, and to study their properties and to divide the microorganisms into a specific groups for diagnostic purposes, we use biological stains and some of the staining procedures in conjunction with the light microscopes that we're using in the laboratory. For this lesson, we will be having our learning objectives. Once you have completed the experiments in this section, you will be able to first is to explain the chemical and theoretical basis of the biological staining, manipulate the techniques of smear preparation, perform procedures for simple staining and negative staining, and describe the method for performing differential staining procedure such as the gram acid fast, capsule, and spore stains. You see here a figure of the chemical composition of a stain. Chemically, a stain is an organic compound which contains a benzene ring and a chromophore and an oxochrome group. You'll see here the stain picric acid is shown in the next slide. So what benzene is an organic colorless solvent. Your chromophore is a chemical group that imparts color to benzene, and your oxochrome is a chemical group that conveys the property of ionization to the chromogen, enabling it to form salts and bind to fibers and tissues. And the two of this, the benzene and the chromophore, are what we call as their chromogens, because they are a colored compound, but they are not stains. So your picric acid, okay, for this example, you have here the benzene, which is a colorless molecule, plus the nitro groups. We have here the chromophore, and it will form a trinitrobenzene, which is a chromogen. It will be yellow in color because of the presence of the chromophores, and then we add oxochrome, and the product would be your trinitrohydroxybenzene, which is a yellow stain of your picric acid. So the ability of your stain to bind on like macromolecular cellular components such as your proteins, your nucleic acids, will depend on the electrical charges which are found on the chromogen portion, as well as the cellular component to be stained. So we'll talk about some of these stains in the very few slides. Your acidic stains. We have your acidic stains, which are anionic. It means that on the ionization of the stain, the chromogen portion will exhibit a negative charge. Okay, these are ions and therefore has a strong affinity for the positive constituents of your cell. So your cell is extremely a blend of a positive and negative ions or constituents, and your acidic stains will bind to the positive constituents of your cell. And proteins, which are positively charged cellular components, will readily bind to and exhibit the color of the negatively charged anionic chromogen of an acidic stain. So structurally, you will see this photo on your right is a picric acid, which is an example of an acidic stain that produces an anionic chromogen. So this is your figure of your picric acid. It's a ring, and then a product of ionization, and it will yield a negative ion or an anionic chromogen plus a hydrogen ion. Next, we talk about your basic stains. Basic stains are cationic, okay, and because of the ionization of the chromogen portion will exhibit a positive charge. It's the opposite of the acidic stain, and therefore it has a very strong affinity to the negative constituents of your cell. So in general, nucleic acids have negatively charged cellular components, and they will readily bind to and accept the color of the positively changed cationic chromogen of your basic stain. So what you see um, on your right is a photo of a micrograph of cheek cells which are being stained by a basic stain. And that basic stain is technically, we use that in that particular experiment, which is a methylene blue. A methylene blue has undergone ionization, and it will be the products of this ionization will become the cationic chromogen, which is a positively charged ion, and a chloride ion. Structurally, you'll see here that the methylene blue is a basic stain because it produces a cationic chromogen. So in general, okay, we have here a summary of your acidic and basic stains. Basic stains are more commonly used for bacterial staining, okay, because the presence of the negative charge on the bacterial surface acts to repel most of these acidic stains, and thus preventing their penetration into the cell. And through the use of some of the simple stains, or series of these simple stains, many attributes of these microbes may be determined. The morphological characteristics and the cellular structures of bacteria can easily be visualized, differentiated, and separated through numerous staining techniques. Okay. For this outline of your staining techniques, we have here the commonly used procedures and their purposes. For simple staining, it must be of the use of a single stain. This is for the visualization of the morphological shape, such as cocci, bacilli, and spirali, and the arrangement if it's in chains, clusters, pairs, and tetrads. For differential staining, and it uses the two contrasting stains. We have here to separate into groups the gram stain and the acid-fast stain. In the visualization of the structures, we have the flagella stain, the capsule stain, spore stain, and the nuclear stain. And we will learn more of them in the future slides. But before that, I have to introduce to you a certain case study that is very helpful in learning all of these microbial staining techniques, which is a initial step in the microbiology laboratory. So here in this case study, we have here a patient presented with technically a disease, an accident. Okay, so during a late night blizzard, there is a patient presenting at a rural medical facility complaining of a possible leg injury. And upon examination of this attending emergency room physician, he notes that there is a deep laceration on the left lateral thigh, and the wound is approximately 60 millimeter in length. Okay, and the assumed infected purulent wound has a greenish pus discharge. So you'll see the pus on the surface and warm to touch, okay, and showing the signs of red streaking extending in his left groin, and these are an indication of an inflammation. So some of the cardinal signs of inflammation is being put up here, and the edges of the wound have begun to exhibit a dark discoloration, which is an indicative of the early stages of necrosis or tissue death. So your case study is: due to the time of night and prevailing weather conditions, the medical center's diagnostic laboratory has been closed and left unattended. And according to the patient's medical history, the patient has shown signs of hypersensitivity to most broad-spectrum antibiotics. Okay. It precludes their use of initially to treat the infection and arrest the potential spread of necrosis. And in this case study, the attending physician is left with two choices, okay, for antibiotics that will treat the patient while not running the risk of including anaphylaxis. So we have here vancomycin for a gram-positive infection and ciprofloxacin for a gram-negative microbial infection. The patient has been treated with both antibiotics in the past. The question is, which antibiotic should the physician use? You can just comment your answers in the comment sections. Thank you. And we will be answering more of that as we progress to this lesson. You have here the molecular structure of the vancomycin for the gram-positive infections, and we have here your ciprofloxacin for a gram-negative on your right, okay, microbial infections. And these questions are being considered. What can the physician do to quickly, easily determine which antibiotic would be the correct one to use for the patient? And would a simple stain be enough to determine the antibiotic use? Why or why not? And I have here an infographic coming from the Choosing Wisely in the US. These are some of the 5 questions to ask the doctor before you take antibiotics. These are the 5 questions. The first one is, do I really need antibiotics? Antibiotics will fight bacterial infections, and like your streptococcus bacteria in your throat, whooping cough, and your symptomatic bladder infections, they don't fight viruses, okay, such as the colds, the flu, and most of your sore throat, sinus infections. And you need to ask the doctor if you have a bacterial infection. Second is, what are the risks? The risk of taking antibiotics would be diarrhea, vomiting, and more. And they could also lead to what we call as your antibiotic resistance. Because if you use antibiotics but you don't need them, they may not work when you need them. The third question is, are there simpler or safer options? Sometimes all you need is rest and plenty of liquid. So the patients are advised to drink a lot of water, hydrate themselves, fruits, okay, and rest. You can also ask about antibiotic ointments and drops for conditions like your swimmer's ear or pink eye. Fourth is, how much do they cost? Antibiotics are usually not expensive, but if you take them and when you don't need them, they may not work for you in the future, and they may cost you a lot of time and money. And fifth and last question, how do I safely take antibiotics? Okay, if your doctor prescribes antibiotics, take them exactly as directed, even if you feel better. If your physician will tell you take the antibiotic every 6 hours for 5 days, okay, you will consume that 5 days timeline and follow the suggested prescriptions by your physicians in taking antibiotics. Antibiotics will help you to prevent or treat some infections, but if you use them for wrong reasons, they may cause unnecessary harm. Okay, you need to talk to your doctor to make sure that you have the right antibiotics for right reasons and the right time. For our next lesson, we have here the preparation of the bacterial smears. And for the learning objective, once you have completed experimentation, you should be able to prepare bacterial smears to microscopically visualize bacteria. In principle, bacterial smears must be prepared prior to executing any of the staining techniques, and this is a necessary procedure. Okay, this is not difficult, although the preparation requires an adequate care. Be sure to properly and carefully follow the rules listed below. And these are the basic principles. First is to prepare the glass microscope slide, label the slides, prepare the smear. It will be different for broth cultures and cultures from solid medium. And for the last step is the heat fixation. So prepare the glass microscope slide. You'll see here a bunch of microscope slides. Okay, clean slides are very easy to prepare, and grease or oil from your fingers on your slides must be removed with soap and water, or like scoring powders. Okay, and it will be followed by a water rinse in a rinse of 95% alcohol. After cleaning, dry the slides and place them on the laboratory towels until ready for use. Note, very important, remember to hold the clean slides by their edges. Okay, you use these edges here to hold the cleaned glass microscope slide. After doing that, you need to label the slides. Labeling and proper labeling of your slides is very essential. Write the initials of the microorganism on either on the end of the slide. You'll see here in the pink slide here, and use a marking pencil on the surface on which the smear is being made. Ensure that the labels does not come in contact with the staining reagents. Very important. Next would be the preparation of your smear. Okay, it is very crucial to avoid thick and very dense smears. A thick or dense smear will occur when there's too much culture that is being used in the preparation, which like concentrates a large number of cells on the slide. And this type of preparation is quite disadvantageous because it is diminishing the amount of light coming through or passing through, and makes it very difficult to visualize the morphology of your simple cell or single cells. In this note, we need to put some small amount, or it requires only a very little amount of a bacterial culture, okay, before we place them in the microscopes. Okay, a good smear is very dry, okay, whitish layer, and the print of your textbook should be legible through the smear. When you try to test it and you're using your textbook, you can just read the text, and different techniques are being used to, okay, depending on whether the smear is made from a broth or a solid medium culture. In future slides, we will see this broth culture. Okay, in your broth culture, you'll see here test tube. It is a broth. It's a liquid culture. You resuspend the culture by tapping. You just tap the tube with your finger, and depending on the size of the loop and the amount of culture growth, apply 1 or 2 loopfuls, okay, to the center of the slide with a sterile inoculating loop, and you spread it evenly. Okay, spread it evenly over an area in about the size of a dime, and set the smears on the laboratory table and allow it to dry. So you use your inoculating loops. For solid medium, okay, cultures from the solid medium, organisms in your solid medium will produce a very thick, okay, dense surface growth and are not amenable to direct transfer to the glass slides, and these cultures will be diluted. Okay, it must be diluted by placing 1 or 2 loopfuls of water on the center of the slide. So put water in which cells will be emulsified. Once done, you need to transfer the cells into a sterile inoculating loop or a needle, and only the tip of the loop or your needle will be used, okay, to slightly touch the culture, and this will make sure that you will prevent the transfer of too many cells. You just need a little. Suspension is accomplished by spreading the cells in a circular motion, okay, in the drop of water, and with using your loop or needle. So you need to have a move it using a circular motion, and this finished smear should occupy an area of about size of a nickel, and should appear as a translucent or a transparent confluent whitish film. You'll see here in this figure. After that, you allow it to dry, okay, completely. Do not blow air, or do not use a dryer or a wave it in the air, because it might affect, okay, the placement of the cultures in your smear. And lastly, we have here the heat fixation. This is a micro incinerator, okay. Unless fixed on the glass slide, the bacterial smear will just wash away during the staining procedure, and this is avoided by using heat fixation, okay, during which the bacterial proteins are coagulated and fixed to the glass slides, and heat fixation is performed by rapid passaging of your slides, okay, into the Bunsen burner, or the flame of the Bunsen burner 3 times, or in front of the micro incinerator as shown in this figure. While many texts discuss using a Bunsen burner for sterilization and heat fixation, the American Society for Microbiology is one of the governing bodies that determines safe laboratory procedures and change the prescribed methods for heat fixation and benchtop sterilization to use micro incinerator. Just a prescribed apparatus instead of a Bunsen burner, to reduce the possibility of aerosolization of the bacteria on the slide or the loop. So you'll see here the next figure are the simplified steps on how we perform this in this stainings, okay, of the broth medium, okay, and the solid medium. This liquid and this is a solid. So again, place 1 or 2 loopfuls of a cell suspension on the clean slide. On the other hand, 2 loopfuls of water on the center of the slide. Next, using a circular movement of the loop, spread the suspension into a thin area approximately the size of a dime. Transfer the amount on your hard solid medium. You need to transfer a small amount of bacterial culture, inoculum for the slant culture, okay, approximately size of a nickel, and allow the smear to air dry. Both of them requires this third and fourth steps in your bacterial smear preparation. And using a micro incinerator, holding the slide at one end, quickly pass the smear over the flame of your Bunsen burner 2 or 3 times. Here are some of the tips for success in the laboratory. The first one will be the bacterial smear should be heavy enough to leave a slight film, but not so heavy that you can plainly see the bacteria, okay, without a microscope, because students sometimes err on the side of adding too much bacteria to a slide to make sure that there are enough, or there will be enough bacteria there for later visualization, and this has the potential to interfere with later staining and procedures and produce sometimes false results. The second will be heat fixing should warm the slide until it's not hot, okay, until it is hot to the touch but not to the point of burning. Overheating the slide during this step increases the potential of damaging the cells, okay, and damaged cells do not retain stains and produce an inconclusive staining results. Underheating also of the slide does not allow the cells to affix to the glass. Resulting washes or stains will rinse the bacteria off the glass, and leaving a few, if any, bacteria present for later viewing. These are just some of the tips that are used for bacterial smearing. We proceed with the simple staining. This is a figure on your background is a Bordetella pertussis, a bacteria that causes a whooping cough. So for this lesson, we need to understand, and once we have completed this experiment, we should be able to perform a simple staining procedure and compare the morphological shapes and arrangements of your bacterial cells. In principle, in simple staining, we stain the bacterial smear with a single reagent. Okay, a single reagent will produce a distinctive contrast between the organism and the background. Basic stains with a positively charged chromogen are preferred because bacterial nucleic acids and certain cell wall components will carry a negative charge, and they will strongly attract and bind to the cationic chromogen. And what the purpose of your simple staining is to elucidate the morphology and the arrangement of your bacterial cells. These are the common morphology of your bacteria. The first is the cocci. Okay, when you say cocci, they are spherical in shape. We have here, accordingly to their numbers, okay, we have here as a Diplococcus because it is a pair of cocci. Streptococcus, it looks like a chain. Okay, Staphylococcus, which is a clustered staphylo. We have the tetrad, which is a packet of 4, and Sarcina, which is a packet of 8. These are cocci. These are bacilli. Bacilli, when you say Diplobacillus, pair of bacillus, rod-shaped, and Streptococcus, chain. Streptobacillus is these are the chains of your rod-shaped bacteria. And lastly, for spiral bacteria, these are the rigid or flexible. We have here the Vibrios. Vibrios are curved rods. Spirilla are helical and rigid. And we have here the spirochetes, which are helixical and flexible. So how do we apply it in the clinic? Okay, for the quick and simple stain, simple stains are relatively quick and useful methods for testing the presence of, determining the shape of, determining the numbers of bacteria present in a sample, and generally involving a single staining step. Simple staining methods are not considered differential or diagnostic and will have a limited use. However, this is a very quick procedure for determining whether a clinical sample has a presence of a foreign bacterial pathogen. So in procedure, this is your simple stain, place a slide of a staining tray and flood the smear with your methylene blue, a basic staining dye, the methylene blue, and allow it for a 1 to 2 minute exposure to your stain. Next will be gently wash the smear with tap water. Next will be your blot dry the slide with bibulous paper. So these are the frequent steps of your simple staining procedure, and some of the morphology and the shape and arrangements of your bacilli and your diplobacilli, the rod-shaped bacteria, your spirilla or your spiral-shaped bacteria. You look here, it looks like a parasite, but it's not a parasite, it's a bacteria because it has a spiral shaped. We have here your cocci. In this micrograph, a spherical shaped bacteria, which is a staphylococcus because they form in clusters. Now, it's negative staining. Your negative staining, you'll see here in the background, is an eosin-nigrosin stain. Technically, it produces a dark background on which the sperm stand out as slightly colored objects. So you'll see in this micrograph, it uses your eosin-nigrosin stain. And in this experiment, we need to understand and meet the objectives to perform a negative staining procedure, okay, and explain the benefits obtained from visualizing unstained microorganisms. And you'll see on this right image here is a nigrosin, which is in saturated solution of your acid lac 2 stain. So in principle, your negative staining requires the use of an acidic stain such as your India ink, or what we call as nigrosin. Your acidic stain, because its negatively charged chromogen will not penetrate the cells because of the negative charge on the surface of your bacteria. Therefore, the unstained cells are easily discernible against the colored background. And the practical application of your negative staining is in 2 steps. This is twofold. First is your heat fixation is not required, okay, and the cells are not subjected to the distorting effects of chemicals and heat. We can see their natural size and shapes. Second, we can observe bacteria that are very difficult to stain, such as your spirilla. And you'll see here in the figure a micrograph which is stained with negative stains. This is nigrosin in your bacilli, using the 1000 magnification. You see the background is slightly dark, and your microbes, okay, your bacilli is highlighted. The surface in the edges is very highlighted in this image. In the clinical application, okay, we need to detect encapsulated invaders when using your negative staining. In the photograph, okay, this photomicrograph will show you a Cryptococcus neoformans that being used with a India ink staining preparation. Life-threatening infections which is caused by your encapsulated fungal pathogens such as your Cryptococcus neoformans have been increasingly steadily over the past 10 years because of the onset of your acquired immunodeficiency disease syndrome and your expanded use of immunosuppressive drugs. And in the clinic, okay, detecting encapsulated invaders uses the principle of your negative staining. Okay, again, negative staining is used to determine if an organism possesses a capsule, which is a gelatinous layer, outer layer that makes the microorganism more virulent. And although it can also be used to demonstrate spore formation, and this technique is frequently used in the identification of your fungi, such as your Cryptococcus neoformans, which is, as I said earlier, is a very important infectious agent found in your bird droppings, and which is linked to meningeal and the lung infections in humans. This is a photo of your photomicrograph of your Cryptococcus neoformans, which is stained by the Grams method. Okay, this is a yeast, encapsulated yeast. So in performing this negative staining procedure, we need to place a drop of nigrosin at one end of the slide. Okay, this is your stain. Next, to place a loopful of your inoculum into the drop of stain and mix it with the loop. Next will be to place a slide against a drop of suspended organisms in a 45 degrees angle, 45 degrees, okay, and allow the drop to spread along the edge of your applied slide. And lastly, you push the slide away from the previously spread drop of suspended microorganisms, forming a thin smear. After that, you air dry the slide. So these are just some of the basic, okay, procedures of your simple stain and your negative staining, and also your bacterial smear preparation. All of these are a part of the bacterial staining lesson presentation. So thank you so much for listening. Okay, in the next video you will be learning differential staining for visualization of the bacterial cell structures. So if you have questions, you can just drop all of your comments, feedback, suggestions in the comment sections below. Again, stay safe.